Welcome to Future Talk. On today's program, we're going to discuss nanotechnology and cellular biology. How can the tiniest man-made machines interact with the smallest units of human life? And what can we learn from that interaction? Here to discuss these questions is my guest, Nick Melosh. Nick is an associate professor of materials science and engineering at Stanford University, director of the Melosh Research Group, and also director of the Stanford Nanofabrication Center. One of Nick's current research projects is nanostraws, which are tiny tubes that can place material in or remove material from individual cells. He's also working on brain-to-machine interfaces, which will enable us to record brain activity with much greater detail than ever before, and that will give us much greater insight into how the brain actually works. Nick, welcome to the program.
Thanks, Marty. Glad to be here.
Nick, how would you describe the broad scope of your work?
Well, what we're looking at is how do we really start interfacing with the fundamental units of biology? How do we try to get a better handle on what goes on inside of us and help cure disease, for example? And a lot of the challenge is matching the size scale and the behavior of those natural tissues. And what we're interested in doing is taking a lot of the really cool technology that's been developed from the nanotech world and then applying it to biology.
Now, one of your big projects right now is the nanostraws, little tubes that put things into cells or take things out. What is the purpose of the nanotubes? What sort of things do you want to put into a cell or take out?
Yeah, it's a great question. So right now, when we have individual cells and you would really like to transform them, for example, a emerging cancer therapy is to actually take your own T cells from your body and train them to actually go and attack the cancer cell in your body. And the difficulty is that you need to be able to deliver a payload of genes inside of that cell to actually express the right markers and do its job. And right now, that's very difficult. And so what we hope to do with nanotechnology, and especially the nanostraws, is to put those genes into the cell without harming it very much.
So you have to get the genes from somewhere and then get them through the nanostraws into the cells?
That's correct, yeah. And so companies have kind of worked out specific proteins that will recognize the cancers. And so you need those cells to be able to express them. And so the critical thing is getting that payload into the cell and getting it to actually express it.
Now, how do you manufacture something like these little hollow tubes? I think we have an image that shows something about the manufacturing process. Maybe we could take a look at that image. Okay, so there it is. So what are we looking at here?
Yeah, so this is what we would call a process flow diagram, and it kind of outlines how we would make this in a nanocharacterization and nanofabrication facility. So the basic idea is actually we take a water filtration membrane. That's that track-etch membrane. So this is kind of a high-end water filter, and it has these tiny little nanopores in it. So if you look at your really high-end filtration systems, you have nanotechnology. What we do is we actually apply a gas phase coating, and this will actually form a glassy-like network on the outside of the surface and into the tiny little pores in that membrane. From there, we actually etch the top layer off and then remove some of the polymer to reveal these little tubes sticking up. And the beautiful thing about that is we can make really large areas of it all at once, and yet they're still nanoscale. Each one is on the order of 100 to 200 nanometers in size.
Now, how do you get that to the cells? Do you find the cell inside the body and get this inside the body somehow, or are you working in test tubes, basically?
Well, the idea with these kind of treatments is actually you take the cells out of the body and then would place them on this kind of platform and transform them and then put them back. The great thing is that you can use your own body cells, so you don't get a mixture of immune response because you're using other tissue. And so the idea, though, is really about getting those materials into your own cells. Yeah.
Now, I think we have another image that shows how the nanostraws put the material in the cells. Could we see that next image, please? Okay, so here it is.
Yeah, and so the idea here is that if I have a cell and those nanostraws are small enough, some of them will actually pierce the cell membrane, which is really tough. That's why we have to go to the nanoscale. If I have something on micron scale, the cells will just sit on top of it. you have to get really small, about 100 nanometers, before it will pierce the cell wall. And that gives you that conduit through the cell membrane and allows you to deliver those materials.
So it's not like one nanostraw going into one cell, but you have a cluster of nanostraws, an array of nanostraws going into an array of cells. Maybe one cell will have several nanostraws. Maybe some cells won't have any.
So we actually have a dense carpet of these nanostraws, and then we'll culture a couple 100,000 to a few million cells all at once. And on average, each cell will be pierced by about 30 to 50 nanostraws. And so those are going to all collectively form your interface into the cell itself.
So the nanostraws will be drawing its cargo from some pool behind it?
Correct.
And some mechanism will be pumping it through the straws into the cells?
Yeah, the interesting thing is you don't have to actively pump it. The distance between the straw itself is only about 20 microns. And so they'll actually diffuse all by themselves. They'll move up and down. And so by that mechanism, you'll have proteins that you put, as you said, in a reservoir beneath the cells will go up into the cell. At the same time, some of the proteins from the cell will go down into that reservoir. And if we're sneaky and careful, we can actually extract that material from the reservoir and then analyze what's there.
Now, maybe cells have some natural defenses and maybe they don't like to be poked like that. I think we have another image which shows some of the natural defenses of cells. Can we see that next image, please? Okay, so the cell wall defense, how do you deal with that? What if a cell doesn't want to be poked like that?
Yeah, they don't want to be poked, in fact. And the cell defense has, of course, evolved over millennia to be very resistant to puncture. And so I'd like to give the analogy, we think of cells as very soft, fragile objects. A better analogy is actually thinking of a plastic bag floating in the ocean or something like that, where if you push on it, you can deform it very easily, but to get through it, to rupture, is actually very, very difficult. And so that's where nanotechnology comes in. By going small enough, it's sharp enough, it can actually create enough force or enough local pressure to pierce through without really hurting anything. On the other hand, kind of current techniques are fairly aggressive. They really have to tear the cell open. And so you often lose a lot of the cells. They will die because of the aggressive nature of this transformation.
So one of the goals, it seems, is to put maybe certain genes into these cells and then put the cells back in the body. So those cells will reproduce and you have those new genes activating. Now, we usually think of DNA as being on chromosomes. If you just insert random genes into a cell, not attached to a chromosome, does that cell know that that contains information that it needs to follow?
Yeah, and so there's a little bit of a black box aspect to it. Once you get the DNA into the cell and into the nucleus, it will incorporate into some part of a chromosome. Now, how highly that's expressed can vary from cell to cell, and so that's something that we have to look at after we've delivered the cargo to see how effective the cells are.
Because I thought that chromosomes are in a certain order, it's a sequence of genes, and the position in that string is also very informational. So if you just take something random and stick it in there somewhere, is it going to be interpreted correctly?
Interestingly, that's how viruses work, is that they insert the DNA randomly at different spots. And over time, we've all developed these viral genomes in our human DNA. And this is a similar process now. Recently, we've heard about the development of CRISPR-Cas9. So this is a new gene editing tool. And that's really powerful in that it can go to very select areas, and you can actually target it to a particular area and put that gene in. And so those kind of tools will be the cutting edge for gene manipulation.
So does CRISPR also poke a little hole in a cell and insert something through that hole?
No, and so that's where we come in. And so we really need to be able to deliver that whole complex into the cell, and we are the providers of that mechanism to get it in.
Okay. Now, what about taking things out of a cell? What would you want to remove from a cell? Is this like for therapeutic purposes, or maybe it's just experimental? You just want to move things around in a cell just to see what happens.
Yeah, this is really, I think, a deep way to understand what's going on inside the cell. So think of it kind of like a blood draw for the cell. So we're going to take a little bit of its blood from the cytoplasm and analyze what's there and tell, is it healthy, is it sick? And we can also follow it for more of the dynamic transformation. So cells might start in a stem-like state and be able to become all different types of cells. And then scientifically, we know we can kind of transform them into different cell types. Maybe they can become a neuron or a heart cell. And we'd really like to be able to control that process. And yet how they do that is still a little bit of a mystery to us. And so by following it with this kind of sampling technique, we hope to be able to measure what's going on in those cells that transform.
Well, the cells decide for themselves whether it's going to be a heart cell or a muscle cell or a blood cell? Or is there some central mechanism that says you're going to be a heart cell and you're going to be a skin cell?
Yeah. So in the body, there's a very tightly controlled environment around that cell that tells it what it should be. It's constantly being reinforced, hey, you should be a heart cell, and that helps guide it. The trick for us is to reproduce that kind of environment artificially. And so that's still difficult. And right now, we're mostly just trying to do it by chemical means. But other people have started to look at mechanics. For example, if it's a heart cell, it should be beating. So if you mechanically pull it, that can help guide it to be a heart cell instead of a neuron, which doesn't have that kind of mechanical input.
So you sort of give it a little external stimulation to push it in the right direction?
Yes, indeed.
What are some of the technical challenges, without being too technical, of course, that you have to overcome? What are some of the difficult problems you have to solve before any of this technology works?
Yeah, it can be very challenging. I mean, the first, as we saw from that figure, is just getting through the cell wall is a big challenge without completely ripping it open, but just a very small penetration. The other thing is seeing what you're doing, right? And so we in the nanotech community have developed a lot of amazing characterization tools where we can look at very small things. But that's usually in vacuum, and cells do not like being in vacuum. And so how do we actually apply some of those tools so we can see that length scale without killing the cells is a real challenge. So just seeing what you're doing.
It must take a lot of very specialized tools to produce these nano objects. Now, do you have to invent your own tools as you go along, or are there existing tools that have already been developed that you can rely on?
Yeah, it's a great question. So there are actually a large suite of tools which exist. They're usually not exactly what we need, and so what we do is we modify them a little bit to actually be able to look at these kinds of systems. At the same time, there have been huge advances in microscopy. So if you've been doing back to high school chemistry, you look through the microscope and you can see very small objects. But people have been able to push that very, very small objects can now be visualized using that kind of system, so down at the 10, 20 nanometer level.
We have one more image regarding nanostraws. Can we see that last image, please? Okay, so what are we looking at here? It's the same picture that we see behind us here.
So this is what it would look like if you were on that forest mat of nanostraws. You would see them protruding from the substrate, and the cells laying down and kind of spreading out on the nanostraw matrix themselves. And you can see some of those would actually pierce the cell wall, enable you to deliver things in or out of the cell. So it gives you kind of a to-scale view of what it would be like to stand on that.
The cells are not all touching each other, clustered, tightly packed. There's space between them?
There's often space between them, yes.
So when will this be used in actual therapy, or is it at that point already?
So we are doing tests with a number of different labs and also with companies now to actually look at using this for clinical therapy and being able to create cells that would have that kind of impact. So it's ongoing now, and I hope in the next couple of years you'll see some products using it.
Now, there's another major project you're working on now, which is brain-to-machine interfaces, which sounds very intriguing. Now, what do you mean by brain-to-machine interface? I gather it's like you're recording brain activity, not necessarily changing brain activity.
Yeah, that's right. And so the idea is, can we listen in to what's going on inside the brain and then communicate that externally? And so that can go to a computer. So maybe you want to move a cursor around on the screen. Maybe you want to move an artificial limb. And all of these really require high fidelity knowledge of what's going on inside the brain to be able to interpret what your intent is.
Now, is the brain capable of broadcasting wirelessly, or do you have to make a physical connection in order to get a signal?
Yeah, that's the trick. So many of you have seen these EEG devices which record on the outside of the brain. Unfortunately, that is just a summation of all the activity in the brain over a very large region. And so it's not that indicative about specifically what you're trying to do, move my hand in this way. So we do need to go into the brain itself to actually record the correct region of the brain to tell us if you're trying to move a cursor or you're trying to move your hand.
So do you have to like go through somebody's skull to make this attachment?
Yeah, and so the idea is that you want to make a small hole in the skull and then be as little damage as possible to go in. And then we want to make it relatively unobtrusive. You don't want to have a large object on your head, but something more like a cochlear implant, which is already a commercial brain device that is relatively small.
So are you actually testing this on humans yet?
No, we're not testing any of these devices on humans yet. We really want to make sure that it both is efficacious but also safe long before we go into humans.
Well, I think we have an image that shows the basic concept of the brain-to-machine interface. Could we see that image, please? Okay, so there it is. So what are we looking at here?
Yeah, this is the idea that we've been developing along with collaborators for the last couple of years. The real challenge for a lot of the brain-machine interfaces is actually how many individual channels that you can record at once. And so typical devices now will record 100 channels, and that's just not enough to really interpret what's going on in the brain and what your intent is.
When you say channel, do you mean brain activity to a certain electric frequency?
Yeah, so in terms of channel, I mean one recording pad, and hopefully that recording pad will record from one neuron. And so it's not the collective activity, it's one of them, and we can see individual firing events from that one neuron. And then we can correlate that with, okay, you were trying to move your hand to the right in that case.
Okay, so can you get to the content, or are you just saying, well, when this neuron fires, then this physical action takes place? So is it essentially a mapping procedure? Which neurons control which bodily functions? Yeah,
it's largely at this point a mapping function where we see this complex pattern. And when we see that complex pattern, that means you're trying to do a certain thing. And we'll see a different pattern, and that means you have a different intent. And that's where a lot of people have been turning to recent innovations in data science in terms of artificial intelligence or machine learning to come up with that mapping of a complex behavior in the brain and then trying to interpret the intent.
Now, once that map is complete, what do you do with that information?
Well, it depends on exactly which neurons you get. So for example, if we recorded from my region of the brain, it wouldn't have anything to do with yours. They're completely different. And in fact, if I just take that implant and wiggle it around a little bit, then it will also be completely different in the signals. And then we have to retrain the entire algorithm to actually recognize it. And that's one of the major problems with today's devices, that they're kind of big and clunky, and they move over time. So you have to do a lot of recalibration and retraining.
So you have to plant something in the brain and it just stays there for a long time?
That's the idea, yeah. And one of the key things is that you'd like to implant it, but have the same kind of mechanical properties as the brain tissue. You want it, if the brain moves, you want your device to move. And right now, they're kind of these very rigid silicon devices that don't move. And so that leads to a lot of problems.
Well, I think we have another picture which shows some of the challenges involved in trying to access the brain. Can we see that next picture, please? Okay, brain-machine interface challenges. So that looks like a little slice of the brain.
Yeah, exactly. And so these lay out a number of the kinds of things that we want to be able to achieve, and yet the difficulties of doing that. And so if you imagine that we have this chunk of brain, as you said, we need to go millimeters deep into that tissue. So the normal kind of nanotechnology that we have these tools for may not be applicable. We need to do something a little bit bigger scale that can get millimeters deep. And yet we still don't want to damage the tissue, so we want them as small as possible.
So you're working with something that looks like a little metal clip which grabs onto a neuron and holds onto it?
No, actually, these are very simple wires. They're micro wires, so they're about 10 microns in diameter, so very small, much thinner than a hair. And yet those can go in, and as long as they're close to the neuron, they can record that local signal. So they don't actually need to physically touch or clip onto the neuron, just to be within about 50 microns.
So is the basic information that either the neuron is on or it's off? Is it basically a bunch of ones and zeros?
Yeah, very interesting question. So when the neuron fires, it fires this, what we call action potential. And so it fires an up-down kind of pulse. That pulse height is stereotyped, as we say. It's very constant. So it's kind of like a 1 or a 0. However, the neurons don't use that as their ones and zeros. They use the firing rate to determine. So how often they fire is basically communicating what they're trying to say.
Okay. And so when a neuron fires, that's giving information, and that information is interpreted to the part of the body that receives that signal.
Right.
And 3 quick bursts means move to the right, and 3 slow bursts means move this way, or...
Yeah, it's not quite that simple, but in terms of, or that clean, I guess, in terms of 3 bursts mean this particular pattern, but it'll be a number of neurons who all fire and all connect and give an overall population average that will actually influence the behavior.
I think we have one more image which kind of shows how this would actually look in the brain itself. Can we see that last image, please? So I'm guessing that those grayish, brownish, squiggly lines on the left, that's the brain?
Yes, so that's the brain tissue, and so you can see this is a relatively small device that will actually go into the brain, into the outer layer, which we would call the cortex of the brain, and then actually have a wireless recording device so that it could transmit the data without having to pierce the skin. And so the idea is that you'd put it inside and seal it up with the brain, and then it would communicate wirelessly outside the brain.
So you wear this and you go about your business and you walk around with it?
Yes, that's
Now,
the goal.
Are you doing animal tests like mice, for example?
Yes, so we've just started doing animal testing to make sure that it's safe and efficacious, and then being able to tell that the system really does what it needs to do when we're preparing for humans.
It would seem like inserting something into the brain could cause some damage.
Yes, and we have to be very careful about that and try to do as little damage as possible, which is one of the reasons that we're excited. By going smaller and smaller in length scale, we have shown that we do less and less damage. And so by scaling to the micron scale, not quite nanoscale here, but we can avoid rupturing, let's say, the blood vessels in the brain, which is a big source of that inflammation and damage.
Now the brain is fairly large, and this instrument is fairly small, so this instrument looks like it could only read the signals from one very tiny portion of the brain. So do you have to implant hundreds of these devices in one brain to get readings over the entire brain?
Yeah, I mean, that if you're going to do the whole brain, that would be quite a task. And so the idea is less doing the entire brain at once, but focusing in on specific regions that are associated with the kinds of behaviors that we're interested in. For example, there's a pretty good map of letting you know where hand motions are or foot motions, and so we know what regions of the brain to target with these kind of devices.
So could you diagnose things in advance? Could you look at a person's brain and say, this person is likely to develop a certain problem over time?
That's an interesting idea. We haven't looked into that yet. We don't have enough data to really be able to correlate those kind of long-term outcomes based on day-to-day readings.
Now, what is the ultimate objective of the brain-to-machine interface? Is it essentially to map the brain, or maybe someday you want to read contents and read people's thoughts directly? Is that even theoretically possible, to know what somebody is thinking by putting an electrode?
Yeah, so right now, not really, no. And so what we do, as you said, it's really a correlative kind of process where we see this pattern and it means this in terms of motion. That's not that we read your mind or know what you're thinking. It's just pure correlation. And so I don't think we can read minds. The end goal, though, is going to be for restoration for people who have neurodegenerative diseases or paralysis or other kinds of deficits that we can correct by actually reading out their brain activity, which is also or often in good shape, and then being able to bring that out to the external world.
So could this tie in with the nanostraws, like certain neurons are defective, so you fix them?
Yeah, that would be a great dream, to go in with the nanostraws together with these kind of probes to be able to communicate inside of a cell. We haven't gotten there yet, but that would be a fantastic vision in terms of watching both the electrical activity and the chemical activity.
Or maybe two neurons are just crossed and they're tangled together. Maybe you could kind of untangle them?
Yeah, I mean, it's a complicated question. We don't know how much the morphology of the tissue really influences the overall behavior. And so even untangling them might not help in what you're actually looking to do.
This sounds like pretty complicated work. How many people work in your lab, by the way?
So we have about 15 people in my lab, but more importantly, we have a large group of collaborators who work together. So we have people from neuroscience and neurosurgery and basic biology working with my team to actually implement a lot of these.
Is there a lot of trial and error, like you expect a lot of things to fail before you find something that works?
Well, we have a general idea of where we're trying to go, and exactly how you get there is a little bit of trial and error, a little bit of guesswork in terms of things that we think might work, and then we see, and then we adjust on the fly.
I think the brain is one of the hardest things to study. It's very inaccessible for one thing, and also it looks like a bunch of gray mush. I mean, how do you figure out what's really going on there? But it sounds like this technology can give some useful clues, leading us further in the direction of how the minds work.
Yeah, we really hope so. I think that there is a lot to be learned from actually seeing the individual actors and being able to see what that circuit is doing, and then being able to interpret that.
So we're almost out of time, so let me just ask one more quick question. How optimistic are you that you're going to accomplish your research goals and we're going to see some results in the not-too-distant future?
Well, we're very optimistic that we're going to get some kind of brain-machine interface going. As we said, that's the long-term goal. Exactly what the package looks like is going to be flexible. We're going to see what works best, make sure that it is non-destructive, and then make it optimal for humans.
Well, I wish you the best of luck, because it sounds like this has a lot of great potential. I've been speaking with Nick Melosh, head of the Melosh Research Group at Stanford. We're talking about nanotechnology and cellular biology and their connection. I'm Marty Wasserman. Visit our website, www.futuretalk.net, and we'll see you next time.